Bro, Witherhoard is always OP.
Yeah, I got the names on. So I introduce myself on the other side. Hello, everybody. My name is Ahmed Bajnet, and you can call me Baj. I'm your class representative for this term, among three others. Siunsha Varghese.
Bro, why does a class
Yeah.
rep have a fucking
And
heavy ass accent, bro?
Mahshat Targati. أقدر أتنفسها. Yeah. So I hope we have a good term together. That's all. Yeah. This is my first term. Yeah. Great, thanks. Do you have anything else you want to add, or? Like there's another one, victory. Like we have three. There's only this one. Oh, there's only three. Well, you can find the updates. So I guess your reps will organize study sessions, and the dates of those study sessions, once I receive them, I'm going to post them here. So right now they're just to be announced. So we'll post them when we get them, and if you have any announcements or anything, just let me know. I'm happy to let you come up at the start of class, or I can put it on e-class, whatever you prefer. Okay. Thanks, guys. Okay. Okay, so today we're going to do an activity day, of course. You're going to need to do your clicker and also a device to submit on CrowdMark. So we're going to do both.
Damn, fuck.
Tonight, deep question 2 opens up. Everyone, final reminder. You must submit to both Critic and CrowdMark. Okay? I think the vast majority of people got it. There were a few that for some reason or another forgot. But, I want to teach responsibility. And you're all adults. So please don't ask me to make an exception and go in and put your answers in CrowdMark for you and everything. That's going to be your responsibility, okay? So just don't wait until the last minute and make sure you submit on both. It only takes a few minutes to submit on each one. Okay? Good. Evaluation stage for deep question 2. So deep questions 2 is due, it's coming out tonight, 7 o'clock. It's going to be due this Friday before midnight. Then the evaluation stage, opens Monday, February 13th, and that's due Friday the 17th. And then we have reading week coming up, right? Yay, everybody. Okay, yay, clap your, I don't know, just let's celebrate. We're all going to be happy for that, right? I know I'm going to be happy.
Wait, reading
for it.
week is not next week?
Yeah, during reading week, you don't need to submit anything.
Fuck.
Chillax, just, I don't know, whatever, study if you want, have fun. Well, you're supposed to read, study for your courses. There will be nothing due in this course, okay? Your feedback component to the deep question 2, that's gonna be due the Tuesday when you come back. And you know that feedback doesn't take very long, right? So, just don't leave it to the last minute, but you have until Tuesday after the break to get it done. All right. Okay, so, yowzers. Okay, so today, student hours, I actually have a meeting I have to go to. I have to leave here probably 1:45. So if you have some questions after class, I have a little bit of time, and then I can make up more time on Thursday. Thursday, you're gonna have the session to work on your deep question 2. Okay, if you have anything else you want to talk about or any issues and you want to talk to me. So today my time is going to be limited. If you can hold it until Thursday, much appreciated. Other than that, I'll try my best to answer your questions before I have to run to one side of the campus and then to the other side of the campus again. Yeah, okay, good. All right, let's begin. So in groups of 2 to 3, there was a little bit of confusion. I got some questions about this, and I know that everybody in the class is not clear on this. So I wanted to have you guys discuss amongst yourselves. So why are you more likely, when you're looking at linked genes, why are you more likely to get crossing over, in this example, between genes A and gene D as compared to gene A and gene B? So look at, you have A and D, crossing over happens more frequently between A and D than it does between A and B. Why? Okay, so discuss them amongst yourselves. If you see somebody sitting alone, wave them over, get them into your group. I don't, 2 or 3, you can have a little bit bigger group than 2 or 3. But anyways, let's have some discussion on this. Okay, great, good. What's the verdict? Somebody want to tell me? Why is this? Why is it more likely to cross over between A and D than it is between A and B? Yeah.
Okay.
Yes, you have the main concept there, and that's what's important. Be careful with describing it in terms of genes, though, because genes, there can be different densities. There is often different densities, like there's hotspots where there's a lot of genes, and then you'll have emptiness, the void, and there's not a lot of genes, and then, yeah. So, but yeah, your general description about the distance, right? The distance between A and D here, it's larger, right? And so than it is between A and B. And so since there's much more distance, nucleotides, there's many more nucleotides between A and D than there are between A and B, it's more likely just by chance that there would be a higher probability of having recombination between A and D, then A and B. Think about it, it's kind of like I used the dartboard analogy. If you had a really huge dartboard and you were to just like, blindfolded spin around and you threw it on the wall, and if you had a really large dartboard, you're likely to hit somewhere between on the dartboard, right? But if you had a really small dartboard, your probability that you're actually going to hit the board are much less. It's the same kind of concept, or even rolling a dice. If I say you're going to roll a die, what's the probability if you roll a die? It's a six-sided die, right? And what's the probability that you're going to roll between a 1 and a 5? Okay, that probability would be pretty high. It's only 6 that's not between 1 and 5. So it'd be pretty high. But if I asked you the same question, I said, well, what's the probability if you roll between a 1 and a 2? Now your probabilities are much lower, right? So it's the same concept. Okay, good. We all got that this time? Okay. All right, so learning outcomes this week, we're going to talk about gene anatomy and gene expression. So identify points at which the gene expression can be controlled. So we're talking about control of gene expression. One of my favorite topics. It's so important. We're going to explain how most cells in an organism can have the same genetic information. So identical genetic information, produce genes, the cells being produced by mitosis, genetically equivalent, but they can have different expression patterns, different functions, different phenotypes, and so on. Okay, so describe transcription and translation using the appropriate terminology and referencing appropriate components. I find, and I'm surprised, I don't know why, but actually it's kind of gotten better, I think, over the last year or so, but generally this transcription and translation, they're very different things, right? So really in your mind, try to separate those two. They're very, very different things. Transcription, the production of an RNA molecule. Translation, the production of a protein. Very, very different things. So try not to get them confused. Okay, so given a gene segment of DNA and its RNA transcript, identify the template, the direction of transcription, and polarities of all the DNA and RNA strands involved. So we go over the basics, and then maybe later we'll get it a little bit deeper. But I remember my training was mainly like given sequences. You look at a DNA sequence, and then all of the stuff we're going to talk about, they would ask you, okay, what's on the template strand? What's on the coding strand? What is the reverse complement here and there? And actually, if you were to do PCR diagnostics in medicine, or if you're to do research and you use PCR or various techniques, understanding that information is critical. Okay? Okay, so we're gonna draw a simple line drawing of a typical eukaryotic gene. So be able to draw a gene, its RNA transcript, identify and describe key structural features, both the RNA and DNA level. So here's your features here, promoters, coding region. Coding region always, often also, so coding region, it can also be referred to as coding sequence, the CDS coding sequence. Introns, exons, termination sequences, and so on, indicate the regions that are genic, so in the gene, but not coding. So remember what we mean by coding. Coding. If we don't say, what does coding mean? If we don't distinguish RNA or protein, what do we say a coding region? Generally, most commonly, what is a coding region? Okay, you know what? I think I'm just going to pull my chair up here and we're going to have a discussion because I got a lot of people here, but it's, yeah, okay. What is the, yeah. Awesome. Yeah, when we say coding region of a gene, usually we're talking about exons, and exons are ultimately what code the protein. So when we say coding sequence, it's coding for a protein. Now, remember, not every gene codes for proteins. That was a big concept that we're learning now, right? Not every gene codes for proteins. We had microRNAs and so on. So sometimes when we say coding region in that regard, we'll refer to the coding of the RNA rather than the protein itself. Okay, but usually that distinguishing, that's given. You know, we'll actually specifically state that we're talking about RNA coding region here. Okay, so transcribed, what is this? Genic, but not coding. So what do we mean by coding? Transcribed, but not translated. So some of the parts of the RNA are transcribed, and even if that RNA encodes a protein, some of the regions are not translated, right? And some of the regions are both transcribed and translated. Okay. FOXP2 is found on human chromosome 7. FOXP2 protein is found in a certain, but not all, brain cells in the same individual who is phenotypically normal. How is this possible? I'll give you a sec to read it over again yourself. Okay. Yeah, 5, 4. Clicker troubles? 2, 1. 205, 206, this keeps going up. 5, 4, 3, 2, 1. Okay. So, one of the things, okay, before I look at the answer here, when we say FOXP2 in italics here, what are we referring to? The gene or the protein? Gene. Very good. When we say it in non-italics, what are we referring to? Okay. Protein, right? Protein. Very good. Now, if we say the protein is found in certain but not all cells, does that mean the gene is found in certain but not all cells? Or is the gene found in all cells? The gene's found in all cells. The protein is expressed in some cells, not other ones. Okay, so let's look at the results we got here. Whoa, awesome, guys. 76% of the class chose D. Very good, very good. So, yeah, so FOXP2 mRNA is only produced in some brain cells. Can be a normal thing, actually. That's expected. You wouldn't expect every gene to be, that's expressed in a brain cell to be expressed in all brain cells. Otherwise, they'd all be the same. Otherwise, if they all had the same expression profiles, they would all be the same, but they don't. Okay, so some brain cells don't have chromosome 7. We know that's not true. They all do. FOXP2 DNA is only present in some brain cells. We know that's not true either. Some brain cells don't have ribosomes. Why is that not true? Some brain cells don't have ribosomes. Actually, only 1% chose that, so we're pretty good there. But why do you think that's not true? Yeah. Yeah, why? Yeah, right. You need to have translation. Very good. Very good. You need to have translation. You need proteins for cells to function and do things, right? Yeah. Good. Okay, so remember I was mentioning before, so how is this possible? You have cells, you have stem cells in the brain, just like any other organ in the body. And ultimately they're gonna undergo mitosis, and that's how they make more and more cells to make up that organ, right? And so mitosis, they're going to be genetically identical. They're going to be the same. And ultimately, let me see here. I actually forgot my pen. Let me see here, draw this. Okay, so ultimately, let's pretend that this cell's giving rise to these cells and here. And then this cell's going to divide again and make two new. And then ultimately, we get more cells and more cells, more cells. And so they're identical, at least in the beginning, they're identical. And remember that when they get pushed further and further away from the stem cell niche, they enter into different environments. We talked about that, right? And what is the consequence of that happening? They may get pushed into a region where they're going to have an extracellular environment, might have hormones localized in it, might have particular proteins that they can sense that are localized there. And by having that different extracellular environment, that may stimulate signaling cascades, it might stimulate phosphorylation of proteins and so on and so forth, that ultimately leads to differences in gene expression. Yeah, so many genes are expressed in a cell or tissue specific way. Okay, so if we look at different cell types, pancreas cell, eye cell, nerve cell, and then we look at these different, like they're just representative genes, right? A gene that's involved in glycolysis, remember glycolysis is like, it's breaking down glucose and using it to produce energy. All cells need to do that. So that particular gene be expressed in all these different cell types. However, some other genes might only be expressed in one particular cell type, the crystalline gene, particularly in the island cells, and not in the pancreas and not in the nerve, right? Okay, so different cell types in the body, they have different expression profiles, different gene expression profiles. And so we're going to talk about how that happens. But, you know, an example that I think of, it could be not just like different locations in the cell. So we got to get rid of that. It's not just different locations in the cell. It could be stress induced. How many of you have midterms coming up? Go. Your gene expressions right now due to your stress are not the same as whenever you're not stressed. There are some genes that are stress inducible, and they're kind of helping you work through this stress, helping your brain deal with the extra activity and everything that's going on. And they may not be expressed whenever you're not stressed. So just a typical example. It's not just cell specific or cell type specific, but it can also be influenced by other things. Okay, so gene regulation also affects the amount of stuff a cell type produces over time. And that could be depending on its environment. So the proteins and so on that are produced. Oftentimes you have a combination, and it's usually like this. It's a combination of where the cell is located, what it's developed into, what genes are accessible for expression, and on top of that layer, we have signaling that comes in from the external environment to turn genes on and off. Those genes that are expressed in one cell type might not even be accessible to be expressed in another cell type. You know, chromatin, condensed, not accessible. Some cell types they are. Are all RNA transcripts mRNA? Oh my goodness, we started off at 100. No. I'm not going to look. Just give you a few seconds. 5, 4, 3, 2, 1. Okay, we're hovering around 80%, so let's stop it there. Or it goes down further. The vast majority of you say no, thank you very much. That's reassuring. Why do we say that not all RNA transcripts are mRNAs? What is an mRNA? I kind of heard an answer. Yeah. Messenger RNA, yeah. And what does the messenger RNA code for? It codes for a protein. Yeah, a sequence of amino acids. Excellent. Right. Okay, so are all RNA transcripts mRNAs? You know that's not the case. You heard about the microRNA. What's another example beside the microRNA? Ribosomal RNAs. Yes, you guys are listening. I love it. Okay, so what is a gene? Remember, it's not all about proteins. That was kind of like a major point. We need to get away from that concept where genes encode proteins. Yes, sometimes that's true. That's right. But a lot of times it's not. Okay, so a definition of a gene must account for encoding other types of functional information, right? And we talked about, where are we in the deep question? We're pretty far along. We're in the evaluation. We're past that. So we talked about the eye locus and the eye locus, how it affected pigmentation. Didn't encode any protein. It encoded a microRNA and how it was able to really dramatically change the phenotype of the seed. And we also talked about how microRNAs were involved in human brain development, right? So really, really important genes. So originally we had a narrow definition of what a gene was, and from the Nature paper that you read, I'm sure you found that even the specialists, all around the world, were having a hard time keeping up, making a definition that fits everything, because new types of genes are being discovered all the time. Okay, so what we're going to talk about is a general structure of a gene. This structure, it holds true regardless whether it's a microRNA. There are features here, if it was a protein coding gene or a microRNA or a ribosomal RNA, there'd be some differences. But this general structure of the transcribed unit being here, and then a core promoter being here, and regulatory promoters elsewhere upstream of the gene, that general structure is going to hold. So a gene is a transcriptional unit, a segment of DNA that contains transcribed region and all sequences necessary to allow transcription. That means that this is transcribed, we're used to calling that a gene, but actually, when we refer to a gene, it has to be expressed. It has to have information that causes its expression. So it also includes these regulatory regions, the core promoter, which a gene needs to be transcribed. That's what RNA polymerase is going to bind. It uses those TAFs, those general transcription factors. I'm not going to make you memorize all. I had to memorize them. Terrible. Because you know what? You know how many of them I remember? Neither do I. I swear. Yeah. So there's no point. There's no but so you want to know though, there are general transcription factors and what they do. They help RNA polymerase get set up on this core promoter. Okay. And that's needed. You need to have RNA polymerase get set up, the DNA unwound, and have that template strand accessible in order for the gene to be transcribed. That doesn't matter, whatever kind of RNA coding gene it is. All right, so, and then you have your regulatory promoter. So this determines where and when. So this is yes or no, I have transcription or not. Here is where and when, in what cell type, in response to what stress. is the expression going to go up or down in response to a stress or in a particular cell type or so on and so forth? This determines where and when transcription happens. And we're talking the transcription way down here, right? All right. So in GEAD only, it includes the transcribe unit, but also the regulatory regions as well. Okay, so in groups of 2 or 3, I want you to have some discussion again. RNA polymerase, a template strand, nucleotides are not the only things required for transcription. What else is required for a usable level of transcription? I say usable level because, you know, nowadays you're probably reading and you like, I think when we described the feature, what is a gene? You had RNA polymerases just going around and transcribing everything. They make little bits of expression of just about everything, but really, to have usable amounts of a gene expression, you need other stuff. Okay, go ahead. Discuss amongst yourselves for a few minutes. What is needed? Okay, great. Who can tell me what else is needed? RNA polymerase, template strand, nucleotides. What are the nucleotides going to be used for? Yeah, to build the RNA. Very good. RNA polymerase polymerizes nucleotides to build an RNA transcript, a polymer. Yes. What other things are required, though, for transcription to happen? Yes. Transcription factors. One of my favorite terms in molecular biology. Transcription factors. Really, really important. What is a transcription factor? Who remembers from Bio 1000? What is a transcription factor? Yeah. Very good. It's a protein. Yes. A protein that binds the RNA, or the DNA. Binds the DNA, and guess what? It binds the DNA, it binds at those, it binds, wait, I think I have a picture there. Yeah, there we go. There's our transcription factors. They're binding the regulatory promoter. Yeah, RNA polymerase brings on some TAFs and stuff that help it open up the RNA, but these ones here are generally what's telling the polymerase the information of how frequently, like when, where, how much transcription to do. Transcription factors, they're proteins, and they're gonna sit on the DNA. I don't really like that, so they don't really do that. They don't sit on, but we'll clarify that in a minute. Okay, so they're gonna be on the DNA, and again, by physically interacting with other proteins, they're determining how much transcription happens. Okay. Where is, there we go. All right. What did the terms cis and trans mean? Five, 4, 3, 2, 1. I wanted to stop it fast because we had 91%. 91% say B. Trans means different and cis means the same. Very good. Very good. it's not just a chemistry term. It means kind of the same thing in chemistry about stereochemistry. But anyways, so whenever we say cis and cis regulatory elements, what are we talking about when we say cis regulatory elements? Yes. Very good, very good. Yeah, and you use both terms. That's great. So transacting factors, transcription factors. Why are they trans? Okay, so let's start the other way around. Cis regulatory elements, they're the elements that they're being bound. That are being bound here. These are being on the DNA. They're elements. They're DNA elements. Okay. Cis means the same. They're on the same DNA upstream of this particular gene, going to control its expression. The transacting factors are the protein. Why do we call them trans? Where is the transacting factor genes? The genes that encode those proteins, the transacting factors, where are they? Yes. Yeah, they're elsewhere in the genome. Yeah, they're encoded elsewhere. They're nowhere near here, but they get expressed in trans from another place in the genome. They come and they bind here once they're translated. So transacting factors, cis regulatory elements. Very good. Okay. What would be cis to DNA that encodes an RNA transcript? Five, 4, 3, 2, 1. Okay. 69% of the class say, 68% of the class say B. DNA sequences on the same chromosome, and you would be correct. Okay. Only DNA sequences, 27% of the class said only DNA sequences in the RNA coding region of the gene, which is no. So, no. The RNA coding region of the gene, so that coding region gets expressed, and actually the cis regulatory elements are on the same chromosome, they're usually nearby, upstream as we've shown in the picture, on the same DNA. Great. Okay, so transcription involves lots of protein things and the sequences of DNA those proteins sit on. Okay, protein things are transcription factors or transacting factors. They act in trans. They're not necessary. They're not expressed from the same DNA as the gene that's going to be transcribed. So they come from trans. And they bind cis regulatory elements. DNA proteins sit on the cis elements. I really don't like that analogy. What are they doing? They're not sitting on. What are they doing? And there's a lot of it. Any suggestions? Better term? Yes. They're binding. Thank you. Binding. Let's just use that term. Binding. They bind the DNA. We're not talking about covalent binding. They're interacting with the bases of the DNA through hydrogen bonds, through van der Waals forces, through sometimes ionic binding. But yeah, so they're binding the DNA. Good. Another way they can also say they're hugging the DNA. They're holding on really tight. Okay, so the transcription factors, they interact with the DNA, and what they do is They do several things, right? One of them is that they're going to interact with RNA polymerase. I even have a little bit of an issue with that because that's the way it's a highly simplified version that they're teaching in this course. It's true the transcription factors can physically interact with RNA polymerase directly, but most of the times they interact through another complex. Does anyone know what that complex is called? Yes, I'll be so happy. Mediator complex. Thank you, sir. Yes, mediator complex, which is kind of like a transducing complex. These transcription factors are binding, they're hugging the DNA, they're binding these particular sequences, and they can interact directly with RNA polymerase, but most often they interact with a mediator complex, which is another group of proteins, and that protein's helping kind of like decode the message. It's helping taking all the messages together so you can have a transcription factor bound here and a transcription factor bound there to these regulatory elements. That's part of the regulatory promoter, which can be very, very far away from the transcription start site. They're interacting together to send RNA polymerase a message. And what they're doing is like they're bending through this complexation. They're bending the DNA and twisting it into a certain conformation that can allow the RNA polymerase to rapidly go through cycles of transcription. You know, if it's held just right, and these are proteins interacting, you can have it set up and then go, set up and go. If you have a repressor protein set up on there, it can do the opposite. It can actually block the interaction of RNA polymerase with that particular core promoter. All right, so yeah, so you have these regulatory parts of your regulatory promoter. These elements can be very distant. So enhancer elements bind activator type transcription factors that increase the frequency of transcription. And if this were a silencer element, it would be a repressor protein that's binding. Okay, so when we think about transcription and what these factors are doing to transcription, they're doing a few things. An important thing that they're doing is they're actually telling RNA polymerase where a gene is located. Okay, because RNA polymerase doesn't know by itself. It doesn't know what to transcribe. It needs transcription factors to tell it where to start transcription. Like there's a gene right here, start here. That's what they're doing by setting up this complex, and they're loading that RNA polymerase in place. Okay, so this is just various three-dimensional diagrams showing this transcription factor, FOXP2, that's hugging or binding the DNA. So you got your double-stranded DNA here, and you have your transcription factors accessing the nucleotides, the bases, usually through the major groove. Okay, they're reading what's there. They're binding and interacting with the DNA elements that are there. And this is just another diagram. There's a three-dimensional, like one of those globular protein diagrams showing the same thing. There's your DNA, and just for you to get an idea about how that all works. Okay. So remember, in diploid organisms such as ourselves, transcription for the most genes occurs from both homologs. Okay, so that means that the same gene can be transcribed, or generally is transcribed, from the chromosome, the homologue that you inherited from sperm, and the homologue that you inherited from the egg. Okay, in diploid organisms. Yeah, so in a heterozygote, there can be two different alleles, right? So two different versions of a gene, and so each found on their individual homologue. And so in a heterozygote, you have two alleles. That would be different in terms of DNA sequence, but both would be transcribed, assuming that the allele is not affecting the binding of a transcription factor, which in turn would affect the expression. Okay, so does that all make sense? That's pretty out there, right? And so, you know, in plants, it's actually very common that you have triploid or even pentaploid, hexaploid, red wheat. Wheat is a hexaploid. It has 6 copies of every single gene. What do you think is going to have higher expression? Something that has 2 copies or 6 copies? Yeah, more copies, more expression. Okay. So how do we identify regulatory regions? Like, how do we know that they're there? We can't just look at DNA sequence and for the most part, like, know whether it's a regulatory region or not. So what people do is sometimes if they think, oh, there's this SNP that we found, that probably is a regulatory region affecting the expression of this gene, this important gene that whenever we have the SNP, we get a disease and the gene's not expressed. They don't know for sure. So what they have to do is isolate this sequence. They need to clone the sequence. Copy the sequence out, usually by PCR. There are various methods. And so we think this is like, this thing is transcribed. We think there's a regulatory region here. So they'll clone that region out that they think is a promoter, and they'll link it to a reporter gene. Who's heard of a reporter gene before? Okay. Reporter gene is a gene that it's much easier to measure whether it's expressed or not than your average gene. And that can be like a reporter gene can be like an enzyme that makes a blue color, like beta galactosidase or the LAX-Z gene. It can also be luciferase, making luminescence, an enzyme. When you mix it with a colorless dye, it actually makes a flash of light. Okay, and it's quantitative, right? You can quantify a reporter gene, how much expression is there based on how much of the dye signal you get. It can also be fluorescent proteins. Who's heard of green fluorescent protein? Yeah, great. That's a reporter. We use it in the lab all the time to figure out where proteins are going. Fuse it to green fluorescent protein and watch where it goes. So that could be like where, but also you can quantify how much. And so people will take these regulatory, we suspect that this is a regulatory element or promoter here that's affecting the expression of this important gene, and they link it to a reporter. And then they take that suspect promoter and the reporter gene, and they'll transfect. They'll introduce it into human cells. It could be a human cell line in a culture dish, for example, and they'll see whether the reporter gets expressed or not under the conditions that they're looking for. Maybe in a diseased cell, they get expressed very highly, and then in a regular cell, they don't get expressed, or vice versa. It can be the opposite. But it's to help them, reporter genes to help them figure out whether those are regulatory elements or not. Okay, so I have an activity worksheet I'm going to open up for you. It's going to be on eClass. open the activity worksheet, follow the instructions. We got about 15, 20 minutes to get this done. Let me open this up here. Groups of 2 to 3. So you're going to submit on CrowdMark. Please don't email me. I forgot to add my friend's name and everything. And they just put your friend's name in the when you do the submission. Okay, so you should be able to see it now. It's week 5, Tuesday, February 7th worksheet. Click on the link, it'll open up a Google Doc. Be prepared to share your answers.
Bro, what the fuck?
Recording stopped. Yeah. Hold on.
I'm just checking that I'm right. Okay, yeah, because I tested this before.
Hey, just a few more minutes. How about another 5 minutes? Is that good to hand in? Okay, if you haven't submitted yet, submit now. We're gonna shut things down in a sec.
Recording in progress.
No. I got some Skull Candy headphones. Now I can listen to Minecraft. That's crazy. My prof is muted. Yeah. It's crazy.
Sure, we can't hear you.
Is it? Don't know. Mm. Because he knows Destiny is a grind. Oh, why's my prof retarded, man? Huh. Only over win is over.